than the wild-type animals, both early in the disease at 3 to 4 months old and late at 9 to 12 months old. And I'm not showing you all the deficits we found in the interest of time, but there's also a deficit in ripple duration. So these ripples are a lot shorter. Instead of being 100, 200 milliseconds, they're more like 50 milliseconds. Now, we also looked at a pattern of activity during the ripples called gamma oscillation. So I'm just going to show you that quickly. This is a ripple shown in a different way. We can take the frequency content of that ripple. This high frequency red dot right here is what we use to detect that a ripple occurs. Down here in yellow is a lower frequency component of that ripple. And if we look closely at the frequencies that are included in there, it's a unimodal distribution around 40 hertz. That frequency is called gamma, or gamma oscillations. It's been studied in many different brain regions. In the hippocampus, we know from previous work that during sharpwave ripples, it's important, or it's correlated with that replay or reactivation. So the better the replay, or the higher the gamma, the better the replay. So we also looked at this gamma activity. And this is showing you the gamma activity in a wild-type animal, and this is showing it to you in a 5XFAD mouse. And the fact that it's yellow here and light blue here means that there's less gamma power in the 5XFAD mouse. Now, if we look at that in a different way, this is showing you gamma power over time during sharpwave ripples, and you can see that there's significantly less gamma power in the 5XFAD. Spiking modulation is also one of the reasons that people are interested in gamma, is because neurons fire at a particular phase of gamma, or they're modulated by the phase of gamma. And so a neuron is more likely to fire at a particular phase, and that means more neurons are more likely to fire together on short time scales. That's important for things like spike transmission and synaptic plasticity. So we also wondered if spiking modulation by gamma was altered, and in fact it is, although the effects are somewhat small. So we're finding that gamma during sharpwave ripples and sharpwave ripple abundance are reduced in the 5XFAD mice, starting even before behavior deficits are detected and continue quite far along with these mice. Now, then we wondered, well, how could that be coming about? So these sharpwave ripples and gamma oscillations, we think, are generated by circuits within the hippocampus that are part of a circuit. I'm showing you a sort of simplified version or a model right here. So a combination of pyramidal cells and interneurons have to work together to produce these kinds of oscillations and bursts of population activity. So for a gamma oscillation, we think what's going on is that these pyramidal cells fire, they synapse onto PV interneurons, and then make those cells fire, and these are inhibitory cells. They synapse back onto the pyramidal cells and shut the pyramidal cells down. So this is a negative feedback loop, yeah? And you can see, this is what we call a raster plot, so you can see spiking activity of the pyramidal cells and PV interneurons. This is from the model, so it's not real data. But you can see the pyramidal cells are firing along, the PV interneurons fire, and then all the pyramidal cell activity stops for a period of time. Now, these PV interneurons are firing together, very synchronized in time, because they're gap junction coupled. So they can basically act like a wave. That means that they can, when one of them fires, it is electrically coupled to the next one and makes it fire. So this produces this kind of period, the cycle where you have pyramidal cell firing, nothing, pyramidal cell firing, nothing. Yeah? So this kind of activity, this PV interneuron inhibition and release of inhibition, leads to bursts of population activity, and if the circuitry is built the right way, you can get cycles of this activity. So we think this is how gamma arises, as well as how you get bursts of population activity during sharpwave ripples. So we wondered if something could be wrong in these relationships, and how these neurons synapse onto each other, or what we call the synaptic efficacy between these cells in the 5XFAD mice. And that can help us understand why these sharpwave ripples in gamma are going wrong. So the way that we can assess that during behavior, so this is, again, we're recording from animals while they're doing these behaviors, we can use what we call a putative monosynaptic connection. So we record spiking activity of multiple cells at the same time, And we can, based on the spike width and firing rates of those cells, identify them as putative pyramidal cells or interneurons. And then we can ask if this cell might be making this cell fire. And basically, you do that by looking at cross correlations between the spiking of these cells. So if this cell makes this cell fire, when this cell fires, this cell would be more likely to also have a spike. And it would be more likely to have a spike 1 to 3 milliseconds after, because we know that's the timing for that spike transmission. So we can assess that with a cross-correlogram. So this is showing you a cross-correlogram where there is a peak at 1 to 3 milliseconds. And this is a cell that, or a pair of cells that we have deemed is putatively monosynaptically connected. And that's because this peak is above the baseline area around it. And then we can measure that connection strength. We take the area that is above a baseline area. Yeah? We can also do that for inhibitory connections. So if this cell turns off this cell, then you should expect its firing rate to go down after the cell fires. Yeah? This is showing you an example of that. If we do a cross-correlation of the spike trains, and you see a dip right here around 1 to 3 milliseconds, that suggests this cell actually inhibited that one. And we can, again, measure that connection strength. Yeah. So we looked at those connections in the 5XFAD mice. And what we find is that there's a significant decrease in this inhibition of interneurons onto pyramidal cells. So that's basically shown here, where we look at many of these connections, average them together. So this is a normal wild-type animal. You can see there's a clear dip right here. That's the inhibition of the pyramidal cell. And then in the 5XFAD mice, you can see basically that dip is missing, where there's a tiny dip. Yeah? And if we quantify that, the connection strength is significantly lower. And so here, everything's negative. So the fact that this is on this side means that there's less of this inhibition. Now, I'm just showing you this during a non-theta period. So that's when these sharp ripples happen. We see a similar deficit during both theta periods and sharp ripples. I'm just not showing you all the data. And we looked at all the different connections. There are some other changes as well that I'm not going to get into, but mostly this is really the most striking one of all the different kinds of pairs that we can look at. So we're finding deficits in synaptic efficacy in vivo. This we think is important because it can help us understand where these sharp wave ripple deficits are coming from. But also, people have long hypothesized synaptic deficits or synaptic loss is a key part of Alzheimer's, especially early Alzheimer's, before there's cell loss. And that's all been studied in slice. So this is helping us to make that link between the synaptic changes that are going on and how that could actually lead to patterns of neural activity that are important for learning and memory. All right, so I've shown you that there are deficits in sharp wave ripples and gamma during sharp wave ripples, and then also in pyramidal interneuron connections, specifically that inhibition of pyramidal cells. And then we want to know, well, what happens if we stimulate some of that activity that's lacking? And I'm going to just tell you right out, we're going to stimulate the gamma activity. And part of that is because we know how to stimulate gamma activity. We have some inklings about how to stimulate sharp wave ripples, but it's not as straightforward right now. So, like I said, this is our high-level overview of how we think about the disease. And then, in thinking about stimulating, we actually thought about, could we be having a different effect and flip this plot on its side, sort of. If we stimulate neural activity, could we be affecting the molecular and cellular pathology of the disease? And the reason we thought that could be plausible is because there's a little bit of prior work that showed you could basically turn up or turn down neural activity and turn up or turn down amyloid. So that made us wonder if we could stimulate this particular frequency, this gamma activity, and also affect amyloid levels. Now, we initially did our stimulation using optogenetics. So optogenetics is a very cool method. It is invasive. It's used in mice. But it makes neurons sensitive to light. And then you can implant a fiber in the region of interest and turn light on and off and make neurons fire basically at will with millisecond precision. So we used a very similar approach to drive gamma that was initially shown by Jess Harden in 2009. And that was to infect these PD-Cre cells. So I mentioned the PD interneurons are important for driving gamma. Part of the reason we understand that to be the case is people have optogenetically stimulated them and found that they can get gamma, yeah? So we optogenetically stimulated those PD cells and looked at amyloid beta levels. Actually, first, let me just tell you, we looked first at whether or not we're generating 40 hertz. So this is showing you this overall sort of stimulation approach, and this is a single trace. You can see before stimulation and after stimulation. There is a change. It might be hard for you to see by eye, but if we quantify that, there's a clear increase in 40 hertz activity when we do 40 hertz stimulation. So this is turning the lights on and off at every 25 milliseconds. And this 40 hertz stimulation is in red. We also did a random stimulation in blue. So those neurons are light sensitive, but instead of turning the laser light on with a 25 millisecond interval, it's a randomized interval. So overall, they're stimulated at the same amount, but it does not produce a 40 hertz oscillation. If you look closely, the blue line is a little bit elevated, all the way from 20 to, I think, even farther out to 60 hertz or so. Yeah, but it's not significant. And then we also do a no stimulation condition where the animals have been infected with a control virus that is not light sensitive, but you still turn the light on. So there's still a laser component. Now we also see that there's an increase in firing of neurons following the light pulse. So we turn the laser on, and then 2 to 3 milliseconds after, we see an increase in firing, both for 40 hertz and random stimulation. So this is simply to show that we can drive gamma by driving PD interneurons at 40 hertz, and we have an important control condition, this random condition, where animals undergo the same amount of stimulation, but it does not produce 40 hertz. So then we looked at amyloid beta levels, and this is amyloid beta 140 and amyloid beta 142. And these are the different conditions I just mentioned to you. So no stimulation, 40 hertz, random, and then we also included an 8 hertz stimulation condition. And what you can see is there's about a 50% reduction in amyloid beta levels following this 40 hertz stimulation. And this was done for an hour. So an hour of optogenetic stimulation is a little bit odd. I'm happy to talk about it at the end. It's kind of a long amount of stimulation. But the other frequencies, random and 8 hertz, do not have this effect. And we also optogenetically stimulated another cell type, and we did not have this effect. So that was surprising to us, and that made us wonder, how could that be happening? So we then looked at amyloid beta, how could changes in amyloid beta levels come about? So amyloid beta levels are a function of amyloid beta production and clearance. And today I'm just going to focus on clearance, and in particular on cells called microglia, because they're of particular interest to me, but also an interest of time. So these microglia have many functions in the brain, but one of them is to serve as trash collectors in the brain. They can literally move around and take up amyloid or other pathogens and break it down and clear it from the brain. So we wondered if 40 hertz stimulation could be affecting microglia. And this is showing you a stain for microglia in green under these different conditions. No stimulation, 40 hertz, or random. I think you can see pretty clearly that there's a difference here. And if we quantify that difference, we find that there are more microglia, they have a larger cell body diameter, and a shorter process length. And these last two are associated with engulfment. That's when these microglia take up amyloid beta. So we also co-labeled for amyloid beta, shown here in red. And when the microglia and amyloid beta are co-localized or overlap, it is in yellow here. And we found that following 40 hertz stimulation, there's a higher percentage of microglia that are also amyloid beta positive. So these were very interesting findings, but we wondered if it was possible that this is really just due to gamma, or if it's somehow some weird side effect of the optogenetic stimulation. Like I said, we have this important control, this random stimulation control, but optogenetics, it's an incredibly powerful tool, but it does actually insert channels into the membrane of neurons. It's probably letting calcium flood into neurons, etc. There are reasons to consider potential side effects. So we really wanted to come up with a different way to stimulate. So one of the ways that we came up with was something called light flicker. So basically, people have shown for decades that you can flicker lights at a particular frequency and drive that frequency in the visual cortex. So we did that with our mice. They were exposed to an hour of lights flickering at 40 hertz or other conditions, which I'll tell you about, 20 hertz, 80 hertz random, for an hour. And for 40 hertz, that's the lights turn off and on for 12.5 milliseconds. And we first confirmed that indeed we could drive 40 hertz activity in the visual cortex. So that's shown here. Again, there's an increase with 40 hertz flicker, there's an increase in 40 hertz power. It's maybe not as dramatic as the optogenetic stimulation, but it's there. And we see neural firing rates go up and down as the lights turn off and on. Now, visual cortex is not that relevant to Alzheimer's disease in general, but in this mouse model, there's elevated amyloid levels in the visual cortex. So we can ask the same questions we just did of the hippocampus in the mouse as a demonstration. And this is just to show you what the actual light flicker looks like with a mouse. And this is actually 20 hertz, not 40 hertz, because 40 hertz is too fast of a flicker for your typical video frame rate. Yeah? But you can see 40 hertz light flicker. All right, and this is, then we looked at amyloid beta levels in visual cortex after an hour of that kind of flicker. And you can see, so this is dark, animals were exposed to dark, light, no flickering, 40 hertz flicker or random flicker. And you can see, again, we're getting this 50% reduction in amyloid beta levels with the 40 hertz flicker. We also looked at several other conditions. So this one here is with picotoxin, which is a GABA antagonist. This is 20 hertz and this is 80 hertz. So it happens specifically with 40 hertz, but not the other conditions. And the picotoxin is a GABA antagonist, so it's showing you that this is GABA dependent. GABA is the primary neurotransmitter of inhibitory neurons. We also looked again at microglia, and we find very similar changes, although there's not an increase in the number of microglia. And we looked at a number of different effects as well. I'll just summarize them for you. So 40 hertz light flicker reduced plaques in older animals, but we had to do it an hour a day for 7 days instead of just 1 hour. It also reduced amyloid beta in aged wild-type mice and another mouse model of Alzheimer's. And it also reduced phosphorylated tau in a different mouse model. Phosphorylated tau is another molecule that we think could be really important in Alzheimer's disease. We don't fully understand the mechanism here, and that's an active area of investigation. So then we wondered, well, how does 40 hertz light flicker possibly mobilize microglia? And this is actually came out of some discussions I had with Levi Wood shortly after I got here. And, you know, so we basically have neurons firing at 40 hertz. We have microglia transforming, and we think that's a key part of amyloid beta reduction. But what happens in between? Well, we thought one of the key molecules involved is likely to be cytokines. Cytokines are signaling molecules between cells. They are well known for their role in immune signaling, but they also have a variety of other roles, including chemotaxis, cell survival, and more. So we looked at cytokine levels following 40 hertz flicker. I'm just going to show you a little bit of that data. So this is cytokine levels following 1 hour of either 40 hertz flicker up at the top, random flicker here, 20 hertz flicker here, or light flicker here. And I think you can see that the 40 hertz flicker, there's just overall a lot more red. That means that there are higher cytokine levels. And each one of these columns here is a different cytokine. So it's really across many different cytokines, although we can break it down by the particular ones. And then each one of these rows is a different animal. So overall, we see a lot more or higher cytokine levels in the 40 hertz group. But that's not the only thing that's going on. Interestingly, 20 hertz seems to be the lowest. Random is somewhere in between. And then light has some of its own effects going on. And this one is not ordered by the cytokines that are most different between the different conditions, but I can tell you that we've done those analyses, and then we can pick apart different individual cytokines and see what role they're playing in these different conditions. So we think that we're driving gamma in neurons that is leading to cytokines that mobilize microglia. And we now have some preliminary data showing that these cytokines are being released by neurons. So that would suggest that the neurons are acting as immune modulators. This is still a work in progress, and we're still working that out. But it is a really interesting idea. I don't think it's unprecedented in the field, but it's not commonly how people think about neurons. We think of microglia as being the immune cells of the brain, and neurons as doing something different. Think of them in different categories. But there are these really important interactions between them. All right, so I've shown you that there are deficits in this gamma activity during shortwave ripples, and then driving gamma reduces amyloid beta and other molecular pathology, as well as recruiting immune signaling and microglia in mouse models of AD. And actually, I should mention that the cytokines that I just showed you were actually in wild-type animals. So we get effects on the immune signaling in healthy animals, not just mouse models of Alzheimer's. Well then, you may be wondering, how could we possibly translate this to humans? Now, one of the challenges here is that first, if we would like to stimulate in humans with Alzheimer's, we'd like to stimulate deep brain regions like the hippocampus. And the hippocampus is particularly challenging to stimulate because it is so deep. So you can see it here in brain. There's a lot of brain on top of it. And typical stimulation methods, you have to stimulate everything above as well as deeper. And your signal basically gets lost as you're trying to go that deep. There are a few new stimulation methods that are under development that can or seem like they could possibly hit the hippocampus, but it's unclear if they could drive it at 40 hertz. 40 hertz is kind of fast for neurostimulation. So we started playing around with different forms of sensory stimulation, and this is work done by Abby Paulson in my lab, who introduced me. And we started playing around in particular, not just with light flicker, but with light and sound. So I should say we looked at the effects of light flicker alone in the hippocampus, and we could not see reduced amyloid and transformed microglia with light alone. So we started playing with flicker and sound. And let me see if I can play that for you. I have a sound sample. Let's see if it works. No? Okay, you're gonna have to mess. Okay, I'll try to pretend for you. It sounds kind of like this. Yeah? It's fast and it's like a texture. People in music theory would call that a texture because the sounds are so close together they sort of become one. Yeah? And so this is showing you neural activity during that kind of stimulation. This is lights and sound together. I can tell you sound alone, at least in the hippocampus, has very similar effects so far. So this is what we call a raster plot. So each one of these dots is a spike. And this is the overall firing rate, so spikes per time. And they're aligned based on when the stimulus is shown, which is shown above. So you can see the firing rate goes up and down with the light and sound going on and off. And this is one example neuron, this is a different one. Now, let me just be really clear, this is a modulatory effect. So it's not that neurons only fire right here and right here, right? The firing rate goes up and down. So we would call that, or we are calling it modulatory, yeah? Now that's actually pretty similar to endogenous oscillations, like endogenous gamma. It's not that neurons fire a single phase of that oscillation. Their firing rate goes up and down along with the oscillation. And we also see very different effects. So this one neuron looks very different than that neuron. And we don't fully understand all of these differences yet. That's an area of investigation. And like I mentioned, we see similar results, at least in the hippocampus, with auditory flicker alone. So we're finding that this 40 hertz auditory or 40 hertz auditory and light flicker can drive 40 hertz spiking in the hippocampus. I wanted to just show you some zoomed in on particular spike trains. So this is one example from way up here, and this is another example from way down here. So you can see that this one neuron can fire in very different ways over time to this stimulus. And again, that's something that we're figuring out what's going on there. Is that because of the animal's behavior state, or there actually plasticity and changes going on over time in the context of the stimulation? Now, those were some examples. We quantify this using circular statistics. So basically, you can put the firing rate on a polar plot like this, where this is the light on and the sound on, and then the light and sound off. And you can see the firing rate is higher on this part, but it's not just a single firing. The cell only fires here. And you can keep the vector response here, and both the length and the direction of that vector. And then we can measure that vector strength for different conditions. So for 40 hertz in blue, random in orange, or no stimulation in black. And we find that there's a significantly higher vector strength for 40 hertz than the other conditions. Although it is still, like I said, modulatory. We can also compute Rayleigh statistics, and again, it's higher for 40 hertz stimulation than the other conditions. Now, the hippocampus is one region of interest in Alzheimer's disease. The prefrontal cortex is another. And you can see the prefrontal cortex here. There are areas that are easy to reach via traditional stimulation methods, but there are areas that are not. So it would be great to also be able to target the prefrontal cortex with this approach. And in fact, this is just showing you an example, but we do get very similar entrainment in the prefrontal cortex. So 40 hertz auditory light flicker induces 40 hertz spiking activity in multiple regions that are affected by Alzheimer's and other diseases. And it's not just 40 hertz, we can also induce other frequencies. So this is showing you an example of 20 hertz and 80 hertz. I want to be really clear here though, neurons don't really want to fire at 80 hertz, so they're not actually firing on every time we turn on this light or sound. They're more likely to fire at a particular phase, but it may be only on a fraction of the sensory pulses. So this is showing you that bar plot here, where with 80 hertz, actually they're only firing on a small fraction of the times we present that sensory stimulus. Overall, this could be an interesting new way to non-invasively modulate spiking in deep brain regions that are hard to reach via other methods. So 40 hertz is of interest to us in particular because of the effects on amyloid beta and microglia, but other frequencies are of interest for a variety of other indications. So it's something that might be generally useful. Now, of course, you want to know, well, does this affect microglia as well? And the short answer is yes. So this is done by our collaborator, Anthony Martorell, in the Sci Lab. And he looked at very similar effects to what I described to you already, amyloid beta and microglia, as well as some additional ones. And this is showing you the microglia following 40 hertz stimulation or no stimulation. And we see similar effects. Now, let me be clear, this is after an hour a day for 7 days, like we did to reduce plaques with the visual flicker and visual cortex. So these were all in older animals. We get a very similar microglia transformation. We also get reduction in amyloid beta, reduction in plaques. And then Anthony also looked at behavior effects, and he's seen some improvement in behavior in these animals. Now, if we really want to translate this to humans, that's great that we can get this stimulation. Again, this is all in mice, so it's yet to be determined if we're going to work in humans. But we also need to be able to read out the effects of this kind of stimulation, not just do the stimulation itself. Now, amyloid beta, there are established methods for measuring amyloid beta in humans. We do that with PET imaging as well as cerebrospinal fluid, and there's new methods like retinal imaging. But we'd really like to know if we're also having similar effects on the immune system as we find in the mice. I think that's interesting in and of itself scientifically, but also if we're having similar mechanism of action in the mice and in humans. So I mentioned to you before, we think that our current model of the mechanism by which gamma stimulation is affecting microglia and amyloid beta clearance is through neurons producing intraneuronal signaling that leads to release of cytokines. So that's interesting at a basic biology level, But you can also look at these cytokines in humans in the cerebrospinal fluid. So it has potentially translational effects as well, or translational implications as well. All right, so I've shown you that there are deficits in sharp-wave ripples, gamma during sharp-wave ripples, and these pyramidal interneuron interactions in the mouse model of AD. And then that driving gamma reduces the molecular pathology of the disease, amyloid beta, as well as I just mentioned in passing phosphorylated tau. And driving gamma recruits the immune signaling and microglia in the brain. And then finally, we can use visual and auditory flicker to drive gamma non-invasively in hippocampus, and we can use this cytokines to detect some of these immune effects in humans. And this is now leading us to some new studies that we're doing in collaboration with others, with human researchers, on these effects in humans. So some of those studies are being done in healthy subjects here at Georgia Tech, some in collaboration with investigators at Emory in patients with Alzheimer's. And those are initially some small studies so that we can figure out what the potential effects are so we can design bigger studies. There's a really big leap between mice and humans, so there's potentially a lot of things that can go wrong, and not just safety and efficacy, but reasons why the same approach that works in a mice wouldn't work in a human. So we need to work through those steps carefully. But what's also really interesting here is that what we're finding is really a novel link between neural activity, and particularly frequencies of neural activity, and the brain's immune cells that I think could apply to many diseases. So just to give you a thought to leave with, oscillatory activity in the brain and immune function have been implicated in a long list of diseases. So this is a partial list. There's varying amounts of evidence for each one of these. Some microglia or oscillations have been, there's a lot of evidence, some there's very little. But all of these could potentially, there could be a role for rhythmic activity or immune function in the brain. And that really leads to that we need a better understanding of how neural activity induces the immune system, either good or bad, and then how can we harness that? And now that we have neurostimulation tools, not just the ones I've shown you, but there are several in the field, what can we do with that information, with that knowledge, as a way to potentially treat or mitigate disease? All right, that is everything I wanted to tell you about today. I'd love to thank my students. I showed you work today from Abby Paulson, Stephanie Prince, and Christy Garza, as well as our collaborators, Anthony Margarella and Hannah Karina and Nila Silam, and Levi Wood, who's been a really great collaborator here at Georgia Tech. I, of course, want to thank my funding and point out that I have a conflict of interest. I own shares of Cognito Therapeutics, which is working to commercialize 40 Hz of the potential therapeutic in Alzheimer's. All right, thank you. Yeah, go ahead.
I know this is really cool for so many reasons. But I guess my quick question is, there's flicker in the light side also. I just wonder, what is that frequency? And how does that affect like humans, or I guess not humans, but any of the mice work? I know they keep the lights on set.
Yeah, so yes, our fluorescent lights flicker. They flicker past what we call the fusion frequency, so you don't see it unless they're broken or not doing what they're supposed to be doing. So we think, but we don't know yet. We think that if they're past the fusion frequency, they're probably not having an effect. But that said, we haven't studied it, and it does sort of bring up a whole host of issues. Or could there be some of the naturally occurring things in our environment, especially since we stare at screens a lot and we listen to things all the time, where this could be playing a role? I would guess no, but we don't know. So that's one. And then you mentioned the circadian cycle. So yeah, the circadian cycle. Okay, so sleep is likely playing an important role in Alzheimer's disease. Sleep is when things like amyloid are cleared from the brain. And there's various studies that have shown disrupted sleep is correlated with things like amyloid deposition or Alzheimer's progression. But it's not, there. It's correlation, not causation at this point. But that said, there's an open question, is some of our stimulation affecting sleep or some of those processes as well? Because we are light sensitive, we mice, all of us are light sensitive, and this weird light exposure could be somehow affecting their circadian cycle. When we're only doing an hour of flicker or less, probably doesn't matter. But when we're doing an hour a day for 7 days, maybe it does, and we haven't studied that. It's something we should, but we haven't. Go ahead.
The quiescence defined as unconscious or conscious of reference?
So when I say quiescent, I mean the animal could be awake and stopped. They're just not actively running or sleeping.
Okay.
We have not looked at any of this under the context of an anesthesia or things like that, although you can see the sharp wave ripples during anesthesia.
I was wondering if ripples represent like dreams.
Yeah, so people think that. So when ripples and this reactivation activity was first discovered, I think some of the headlines, this was decades ago, 90s. Somebody said, I think one of the press headlines was like, you can see mice dream or rats dream, because that was one of the first potential indications. The ripples happen in particular during slow wave sleep, whereas we think of dreams, or at least the most vivid dreams, happening during REM. So maybe not, but we can't ask the rats what they dreamed about. Yeah. Sort of a problem in general with rodents. Yeah. Go ahead.
Very interesting research. So for the auditory sleep, you need the sound. It's not very friendly, right? So we all use music to sleep it. But there already some study.
Yeah, that's a great question. So we're working with a professor in the music department on making a version that would be pleasant. Yeah. And I don't think it's that unpleasant, but if you have Alzheimer's disease and this is your treatment, it's not that bad. But if you don't, maybe you would want to use this as a preventative. It's not, you're right, it's not that pleasant. And the interesting thing about working with a musician is she understands music theory. Like, I think about it as a neuroscientist and things turning off and on. She's thinking about the sort of long history of how these different signals have been incorporated in sound. It's a very interesting process. But yeah, I don't know. We don't know what the effects are yet.
Also, no report in the literature.
What was that?
I mean, no report in the literature about the music stimulus.
There are reports in the literature interestingly, about using music in epilepsy to reduce seizures. But I don't know that people have looked at amyloid. There's one study that I know where people did, actually I'm not sure if that's published or not yet. So yeah, I'm not sure. Yeah, go ahead.
And then you teased me, but then you took it the other direction. You took
Yeah.
the red pill to the blue pill. So you had the one diagram about enzymes that degrade amyloid beta plaques. So I love proteases. So what has the literature shown about proteases that they've been identified that are up or down regulated in Alzheimer's, and that they don't actually affect that?
Yeah, I wonder if I should go back to that slide. Yeah, so we did find, you mean the amyloid production side, yeah? Like how we go from, when I first brought up microglia, is that what you're?
Yes. Right, the left side.
Yeah. So, so we looked a little bit at amyloid beta production. Basically, the cleavage of APP to get amyloid. And there are a couple different, like beta secretase and gamma secretase are involved. We could talk more about it. I haven't pursued it that much because I think that the microglia part is, right now in the field of Alzheimer's, I would say there's a debate about what's really the underlying pathology. And amyloid has been the thing for a very long time. There are people who still absolutely believe that it's amyloid. But there's now growing evidence that microglia are a key part of it. and that's based on a variety of genetic studies. So there's genetic evidence for both, but I think personally the microglia are really interesting in the context of Alzheimer's, but also potentially play a role in many different diseases. So I haven't focused myself much on the amyloid beta production, but I'm happy to talk to you more about it. Go ahead.
Have you explored at all the intersection between the 40 Hz waves and gamma in terms of making of sound and the idea that they're all beats, which also have those two sounds and the difference
Yeah.
between being that 40 Hz actually is a little more pleasant than the MRI
Yeah.
machine sound?
Yeah, that's a great question. So somebody has brought up to me that also people use it for like meditation and things like that. So apparently there's this 40 hertz kind of stimulus that people have been using for a long time as part of meditation or other focusing kind of activities. We haven't explored it at all with the mice, but it's a great question. It's something we could potentially try out.
And then the other thing that's interesting about it is there's a soft science area, EMDR, regarding post-traumatic stress disorder therapy and other related therapies that specifically uses the same level of stimulation, either in a handheld device or in oral stimulation.
The binaural.
The binaural means, or they use a stimulation device that's called a command.
Oh, interesting.
And somebody that is undergoing that therapy is called to recall a traumatic event while undergoing a processing. And it anecdotally, because I don't know that there are any of these running up anecdotally, it is said to literally rewrite the experience of that trauma in the person's memory when processed and having the exposure to that level of gamma and the hertz at the same time. So it might be an interesting area.
Yeah, no, I didn't know that. Cool. Other question? Yeah, go ahead.
I'm just curious kind of on the timeline and the next step from animal models to human studies.
Yeah, so we have a human study with Alzheimer's patients going right now at Emory. It's a small study, so the primary outcomes of that study are what we call safety and tolerance. But we are also measuring a bunch of biomarkers that could potentially help us understand what to do in the next larger study. There are also some other studies going on from Cognito. I'm not quite as familiar with where they are exactly. So it'll be certainly a few more months before we have results that we can make sense of with our study. But I think, the reality is it's going to be years before we can conclude anything. And, we are those of us who are doing it, we're all really prepared that like the first results might be ambiguous because there's a lot of decisions we had to make to get to this first study. So far the safety and tolerance have been good, right? That's the good news. But is it going to be enough stimulation or not? Do we need to be stimulating? Do we need to be reading out? We tried to include a number of different readouts, and hopefully one of them is a useful one. But there's a lot of unknowns still.
So when we talk about Alzheimer's patients that you study, how do you know they're Alzheimer's patients as opposed to some other form of dementia?
Yeah, that's a great question. So the patients that get included in our study are considered they have mild or early Alzheimer's, and that is based on a neurologist assessment that includes looking at amyloid and tau levels and in particular a ratio between the two, as well as they have to have mild memory impairment, and they have to exclude other possible reasons. So the Alzheimer's group at the Emory Brain Health Center feels pretty confident about their designation, but those patients are going through a large battery of tests. Your average person going to like their primary care physician or their neurologist is going through probably less evaluation. All right, thank you very much.